question that she wanted to know is she said, what advice does Russ have for me to be as successful in my career as he has been in his?
I'd say two things. One is embrace the fear, right? You will not make it in this field unless you take a chance. You'll be happy, maybe. You'll be compensated, but to get that level of happiness and to stretch. And I remember after being at Bristol Myers Squibb for 18 years, the hiring manager at Merck said, "Embrace the fear," and I'm like, "All right, I'll do it." And I tell you, I've never looked back. Moving over.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hi, everyone, and welcome to BioTalk Unzipped, where we unzip the stories behind medical progress in a fun and intriguing way. I'm your host, Gregory Austin. I'm here with my good friend and co-host, Dr. Chad Briscoe. Say hi, Chad.
Hey, how's it going, Greg? I'm looking forward to another recording today.
Absolutely. So this is episode 3. And in this episode today, in our online studio, we have our good friend, Dr. Russ Weiner. And Chad, would you be so kind and introduce our very first guest?
You bet. It's my pleasure to introduce Russ, one of the really big movers and shakers in the pharmaceutical industry. Russ has worked in several senior level roles in the world's leading pharmaceutical companies, such as Bristol Myers Squibb, Merck, Daiichi Sankyo, Gates Medical Research Institute, and currently Takeda. He has over 30 years direct hands-on experience in pharmaceutical and biologic drug discovery and development, clinical trial conduct, and project leadership. He has extensive experience in biologics, biomarker, and companion diagnostic assay development. It's a mouthful, even for those of us who talk about this stuff all the time, Greg. He has participated in the validation and deployment in global phase 1 through 3 clinical trials. He's responsible for building and maintaining high-performing regulated bioanalytical GLP and clinical teams. He's involved in molecular biomarkers, companion diagnostics, and bioassay operations in his various organizations over the years. He's led early clinical development teams responsible for transitioning multiple small molecule immunology inflammation therapeutics from discovery through phase 2 proof of concept. He was directly involved in the submission of over 30 INDs supporting company-wide therapeutic areas, including, but not limited to, oncology, rheumatoid arthritis, Alzheimer's disease. And this is the big one. If you're in drug development, being involved in actually launching even one drug is a huge accomplishment in your career. But Russ has 6 here, I think. Erbitux, Orencia, Nulogix, Yervoy, Keytruda, and its associated companion diagnostic. And I'm going to read it because this is a mouthful for all of these non-pharma folks out there. It's associated approved DACO, PDL1, IHC, 22C3 pharma diagnostic companion diagnostic assay. And if that isn't enough to keep a guy busy, he's also involved in various roles over the years in the AAPS, American Association of Pharmaceutical Scientists, where he's currently president-elect. He's governance board member of Fight Global Cancer, and he is past and current member of several corporate boards. Wow. Welcome, Russ.
Wow. I don't know what to say, but thank you for having me here. It is long, and that just means I'm old.
Well, we appreciate it. Russ is also, I've known him for years, a delight to talk to and a lot of fun. So we'll kind of jump right in and get to know you a little bit, Russ, and share your background with our audience. So what got you in to this roller coaster of an industry? What made that happen for you?
Yeah, so when I was deciding what to do and I went to college, it wasn't too hard. My grandmother had passed from multiple sclerosis. And so I was driving my career in some way to do multiple sclerosis research. But long story short, I was recruited to do a bioanalytical role. And fortunately, the first drug I worked on actually was for multiple sclerosis. It really didn't
Wow.
go anywhere, but it really did allow me to see what the patients are going through and how challenging the disease is above and beyond the personal contact. And so from there, that's where it brings me today, focusing mostly on cancer. But as the intro said, there's a number of diseases, whether they're transplantation, rheumatoid arthritis, mostly in the autoimmune type or immuno-oncology type of drugs. And my, recently moving to Takeda, actually this week was my 2-year anniversary there. And the goal here was, I have at least 1 or 2 more drugs left in me. And
Nice.
hopefully we'll be able to do that in the next few years.
Fantastic.
Yeah, that's fantastic, Russ. I love that. You got 1 or 2 more drugs left in you. That's good. It sounds like it's like Bill Belichick, right now. he may not be coaching at the Patriots much longer, but he's got a few more seasons left in him somewhere.
Yeah.
Exactly.
But hopefully you're doing 1 or 2 more drugs at Takeda.
No, I love it. The idea of seeing these patients, and for those who have not been in drug development long or have gotten far enough along where the drug is actually very close to or is on the verge of being approved, what we usually do is bring in a patient or two. And I can, be proud to say that I'm a crying audience member watching, patients come in. Worked on one project where someone walked out of hospice in an oncology trial, things that you just don't see in your life, and really, allow you to understand the challenges that these people are going through, and to see someone come out of a wheelchair after years and years in a wheelchair. But the hospice one, we haven't found another example of people leaving hospice in stage 4 cancer. So it's quite remarkable.
That is amazing.
I think that might become more normal, though. I mean, let's hope, right?
Mm-hmm.
We might need to change the definition of what hospice is
Mm-hmm.
if we're really successful and can make that more normal. People who think they may be at the end aren't.
Yeah, I mean,
That's what it's all about.
in immuno-oncology, the problem is everybody's immune system is just a bit different. And so this individual went in, looked like the drug didn't work, and then just kicked in. His immune system just got to that point where there was enough response. And I agree with you. I think you're going to see it, actually quite a bit, or people waiting a little bit longer before they give it up. I think these immuno-oncology drugs, you really need to take some time and let them do their job.
Very good. I volunteered for a short while at a hospice center, and that was 10 or so years ago. And people check in, they don't check out.
Mm-hmm.
So that's a remarkable story. Thanks for sharing that.
Sure.
Very good. And how have you seen the industry really evolve over the past 30 years that you've been involved in it?
Well, when I started, biologics were not a thing. And in fact, the CSO of the company didn't believe in biologics. And so we spent a number of years
Wow.
as the poor stepchild, trying to get resources, trying to get SOPs written for biologics. So for the first part of my career, everything was mass spec. You made it fit. You hoped that it worked. And then as we had, again, those of us that have been around, seeing the Crystal City type meetings, the early days of, draft guidance that ultimately led to, I think it's 2018, the most recent guidance that came out. And it's now separate worlds. But fast forward, when I couldn't believe that anybody would want to take a large molecule and put it on a mass spec, now everybody does it. It's actually standard. And I think that's one of the greatest changes is watching chemists, become biochemists literally overnight as they hook up and they interact with individuals from the other side, the dark side, the biologic side. And now we're all on the dark side. So it actually changed quite a bit. The other piece is I find that the bioanalytical staff has changed so drastically as companies have cut back, and those individuals went into the CRO space and really became, stellar individuals there. But what has happened is, as people went to the CRO space, they don't have as much drug development experience. And so it's a real challenge, in the hiring space where you can find excellent bioanalytical chemists all over the world now. But to get them to come in and understand how to function within a drug development paradigm versus a got to get this done, get it back to the customer, and really not understanding the biology behind it, that has really been the most drastic change that I've seen in trying to get people in. So it used to be bring people in and teach them a little bit of regulatory, some GLP, and now we have to teach them, drug development. Once they get that skill set, the biggest challenge is keeping them employed, and so hopefully they don't leave because now they're a rare commodity.
Wow. That's really
Yeah,
interesting.
that's really fascinating because on the flip side, having been a career in contract research, we bring folks over sometimes from pharma, and as you mentioned, it's been kind of more common recently, and you see a similar but different adjustment to go the other direction, where we can't focus as much on the biology and worrying about some of the very important details. We need to leave that to the pharma experts who know the drug and know the biology better. Whereas we need to get the analysis done. We need to crank it through. We need to hit timelines, all while maintaining good science and good quality. But it's really going both ways now. That's interesting because I hadn't really thought about that side of the challenge, I guess, for us. So that's
But
interesting. Yeah.
and as those people interact with their sponsors, having that drug development experience allows them to understand why a sponsor may be cranky, why they need something so rapidly. In cell
Yeah.
therapy,
Yeah.
you get an AE in cell therapy, you want to know what is going on, right? And so normally when there's an AE, particularly in early development, what happens? It's always the assay. You got to check, what's going on. Is it an immune response? Is the drug at a concentration that is well beyond what you expected based on the pharmacokinetics? And so we have to be on point to get these things out. And it's helpful, when individuals really do understand each other and work more as a partnership versus the CRO versus sponsor. So I always keep them close. I just, see my CRO partners as an extension of my team.
Yeah, that's cool. That's cool.
Nice. What do you think, Russ, has been maybe one of the biggest challenges for you, working in this industry, especially on the biologic side?
Pre-analytics. Everything that happens before it gets to the lab. Once it gets to the lab, I'm pretty comfortable. I mean, my assumption is we got good scientists doing good work. We pick good labs, but we can't control what happens at the site of at the study level, in the clinical site. We think we can. We provide instructions, training, investigator meetings, but when all is said and done, it's really up to the site personnel. And in a post-COVID world, it's gotten pretty challenging. It was challenging before, but now it's really challenging in that people move around a lot. Working and training either a nursing staff or technicians to do sample prep, whether it's to prepare those samples for flow cytometry, whether it's to prepare those samples, for an LCMS assay. Maybe they need to do something to stabilize the samples. You need some skills, not just nursing skills. You need some basic lab skills. And once you train a site, that's not the end. That's just the beginning,
Mm-hmm.
right?
Mm-hmm.
And 6 months later, you got to see who's there. You got to see if you need to train. So we require any personnel change to be brought to our attention immediately. And then in a post-COVID world, getting those samples around the world, really challenging. So when I was working at Bill and Melinda Gates, getting samples from all over the world through South Africa and through other countries in a time where flights are canceled. They have not come back. The number of flights, the ability to get those samples around, the ability to get dry ice. Some countries don't have dry ice, and so we're a bit spoiled in the Western world. But that pre-analytics, the ability to get that sample collected, processed, shipped, and stored is still the largest issue we face all over.
Russ, I just had a conversation about this today, so it's really interesting you bring it up, because we're saying, hey, as a bioanalytical lab, we know our challenges, and I'm not, not every one of them are solved, right? There's always those challenges, but we're saying, boy, our customers, by the time they come to us, they're comfortable with what we're doing in the lab, but what we hear from them is, man, how do we make sure it's collected the right way? How do we make sure it gets in the boxes? How do we make sure those boxes get to you in a timely manner? Like we were talking about flow cytometry samples. How do we make sure those really get to you in under 24 hours when that's all the stability there is there? And so we're looking at, how do we find different partners and experts in this space that can help us do it? And then when we get the samples, if there's 1,000 samples, there's like 50 or 100 discrepancies that we have to sort through, and that's just a nightmare for us. So how do we solve all those problems? So it's interesting, as much as the science is, the science is the fun part to solve, but we kind of have our arms around the science, and you're saying the exact same thing. We got our arms around the science, not that it's easy, but yeah, it's these other distractions that are harder and harder. Do you think that's a post COVID world? Do you think that's the development of the new therapies we have? Why do you think that sort of has changed of late?
So in the flow space, because the world, since Yervoy, Keytruda came on the market, there's a tremendous push. Everybody has immuno-oncology now. Every other day you'll see some sort of trade journal or news report about another immuno-oncology drug or immunotherapy in general. In order to understand the biologic or the mechanism of action, there's a tremendous amount of flow. The world is not prepared to do a tremendous amount of flow.
Mm-hmm.
The world is not prepared to do a tremendous amount of high quality flow. So you get flow done, doesn't mean it's high quality. And so we spend a tremendous amount of time on whether it's proficiency testing, working with the vendors, subject matter experts from the company, working with subject matter experts. But it has gotten progressively more challenging because of this shipment issue, the move to do more studies in Pan Asia and in China, and to set those labs up to do high quality flow, whether it's just staining the samples or whether it's preparing PBMCs. How do you get those samples to a processing lab within 6 to 8 hours if you're doing functional assays? And what teams don't realize is your typical less than 10 color flow. Those are a thing of the past. Not really a thing of the past. We still do that, but we're in high dimensional 30 color flow, and any permutation, any change just messes it up. So for example, you got lots of different cells in that sample. What's the stability of your particular sample? And if it went out of stability or the temperature went out of range, is your cell affected? Are the other cells affected? And so just because you got a nice, well-controlled environment, in the lab, as soon as you go out, it's the wild, wild west. So I think COVID has just made it worse. I think going into immuno-oncology and the volume of flow has made it worse. But the bottom line is, just because it's worse doesn't mean it's bad. It just means we have to think harder, have to work harder, and have to find new ways. And whether it's attacking the biology of those samples to extend the stability, you mentioned 24 hours, other assays for 72 hours, and some people try to push it up to 96 hours. Without some breakthrough soon, we're going to be doing these little studies. And so what I've been encouraging people to do is don't do everything at every site. Some sites are better than others. So for example, if it's a challenging flow assay or it's an assay that the lab staff or the hospital staff need to do something, even if it's a simple pipette, don't assume, everything works, perfectly, particularly with those new people that may have come in.
You talk about the challenges of flow. We actually and going above and beyond in the pre-analytics or the outside of the analytics. So last winter we were running a study in our Lincoln lab, and sure enough, we had an assay that had, I'll say, zero stability. I mean, I think we said it had like 2 or 3 hours, but we're carrying it from the clinic right to the flow cytometers. It's right there in the same building. So
Yep.
but of course, on that day, there's a massive snowstorm. All the participants are in the building, and so they're able to draw blood, and they got, but hey, guess what? The guys that run the flow cytometer are snowed in, right? So it's like, all right, who's got a 4x4 pickup? They can go and just pick up a couple of these lab analysts, that can run the assays. So we had 4 people trained, and they're all snowed in, or can't get in. And so literally one of the managers in the clinic said, I'll do it. We're in Lincoln, Nebraska, right? So this is nothing, right? So they go out, they trudge through the snow, and they pick up a couple of the lab analysts and bring them in so they can run these samples, because that's the world we live in, right? And it was a matter of not, and it's not just saving the sample, right? It's okay, great, you saved the sample, but it's about, you brought in a participant, you drew their blood, you're trying to develop a drug, you're trying to move the study forward. Losing a couple of samples, I mean, that it probably wouldn't have crashed the study, but it would have made it less informed, right? Less useful, and it can delay getting one of these therapies to market. So, anyways, it was just, that was a crazy story, but it was funny that it happened, because I actually did that earlier in my career, about 10-15 years ago. I did a similar thing, not for flow, but just to keep the samples moving through the lab on a snow day. So it happens. It's
Well,
reality.
this is the time of the year that we all get a bit
Yeah.
nervous. We're getting into the winter season, and we watch the news and the weather very carefully.
Yeah.
And it will be a challenge, particularly if you lose that baseline sample. You can have all the other samples, but if you miss that baseline sample and all your calculations are percentage of baseline, you're toast.
Do you think communication has improved? Because, again, thinking about this post-COVID world, and we need better communication because of all these challenges we've been talking about. Is communication better now? Are people more comfortable making calls or reaching out, or is it still the same challenges that we had in the past to talk about these things?
Yeah, so that's another thing that has changed over 30 years. It was you'd send it out, you get your data back. Assays were a bit more simplistic. Now we have individuals on the team, whether they're subject matter experts on immunoassays or mass spec, but we have assigned individual subject matter expert for every single CRO that we work with. We do regular touch base, lots of hours spent there that, years ago you didn't. You just assumed everything was good. But the data is so complex that if it does not make sense, if it does not follow the appropriate controls, I mean, we have examples of assays that, I'll go back to flow for a second, where the gating was a bit off, and I'm like, oh, that's not going to work real well. Or a sample comes in and you're wondering what to do with it. Why? Because it doesn't have a label on it. Why should samples have labels on them? So all these type of things allow this individual to have that very close conversation with their project manager or however lab director. And we spend, I'm not going to say inordinate, but it is a ridiculous amount of time to make sure both parties have what they need. Last thing we want are finger pointing. Oh, you didn't get your sample to us. Well, you didn't tell us you were shipping the sample to us, right? Those type of things are a thing of the past because we have these active meetings with each and every CRO on a regular basis, some weekly, depending on the importance and how fast things need to go. As I said, cell therapy, once you find that patient, they're in the queue and they don't have time for a mistake. And so as you mention it, you have to do it in real time.
Pulling out all the stops.
Absolutely. And so, Chad, you had a follow-up question with regards to labs and Russ's types of role that he chose.
Yeah, Russ, I've read it in the biography, and you've alluded to it a couple of times. You've always been a builder. When I think about watching you jump through a few different roles in your career, it's like, whether it's the first to set up new technology, or you're asked to set up a lab, but you don't just set up a lab like the other pharma company has it. You do something new and innovative, always at the front. I really consider you a transformational guy. I guess I'd like to understand what drives you personally to be that sort of a transformational individual.
So my passion is actually, believe it or not, management. The science is the passion, but watching, young scientists, you get them when they're young, you work with them, and all of a sudden they're your boss. I don't know what happened. I'm reporting to someone that I trained, but that the ability to keep that staff means you have to do something different, right? Why are they going to stay with you? Part of that is I love beta testing. I love getting that new toy in the lab. I'm building labs, right now. We just bought all the equipment. It's just like Christmas. It's amazing. And now everybody's excited. They all want to set those new pieces of equipment up. Yep. So, whether it's new pieces of equipment or finding areas that make people crazy, right? Get rid of the friction points that make people crazy.
Yeah.
And if, whether it's, as we just spoke about, having someone talk to the CRO, so someone who's more of a lab rat, don't bother me, just let me do my work, does not have to get out of the lab and get into those conversations, which they don't want to do. Identifying areas for career growth. And the labs are set up in such a way that we give people a tremendous amount of flexibility, a tremendous amount of, the ability to almost hang yourself. So the labs are built, with a very high level, very, good people who know the space, but whenever we build the labs, we want people to take chances. And the only way you take chances is if someone has your back. And that's where we see the biggest breakthrough, whether it's technology, ways of doing things, the ability to do things faster, better, cheaper, but of acceptable or higher quality. That's the way I build labs. And I'll tell you, nothing better 30 years later than I see applications that were built at my prior companies. They're still using them. So even though we got this new technology and AI, we're using stuff that's 20 plus years old and it still works. Who knew? But that type of doing things just a little bit differently versus snap it all together, put it there. This is how all companies work. And in fact, I'd rather stand back and bring these people in and let them tell me, what's wrong, what doesn't fit. They can call my first child ugly. It's that first validation report, that first child's ugly. And you don't take it seriously. You don't take it not seriously. You don't take it to heart. You're fighting the science, not the person. And to get that lab, that environment of people helping each other, the science comes. They all come in with the science. But can they work together? And the example I often give is if you come into the lab and people are sitting there and someone has their head down and they're trying to figure out what life is all about and they got an issue and some clinical team got in their face because something was delayed. Are the other people in the lab, are they the type of people that see someone struggling and go over and ask to help? Can I take something off your plate so that you can focus on this? Or are you turning and saying, no, that's not my problem? And so that cohesive way of having people care for each other on the personal level is really what keeps the attrition down, the interest up, and the curiosity just flows. And the one of the most, one of the things I'm most proud about is the fact that the attrition rate in that 30 years is fractional. It's not even at a 1% per year. Some would argue that's not good. You want to get new blood, but I don't like recruiting and training, people. I'd rather they stay forever. And in fact, some of the companies, the people that I initially hired are still there. They're lifers. And nice. The beauty is they just make you so incredibly proud. You mentioned, PharmSci 360.
Russ, are you getting choked up? It looks like you're getting choked up.
Well, I'll tell you the choked up story.
No, that's all right. That's all right. That's awesome.
I think it was Rib,
Tell us.
the last meeting, and had a colleague that I hired that I remember nobody wanted to hire this individual, but I said, look at the upside potential that this individual had. No one on the interview team supported it. I was the hiring manager. We had a policy, hiring manager wins, brought that person in. And at Rib, that individual sat next to me on the stage in a panel discussion. Then I choked up watching that individual's kids grow up from a postdoc to now a leader in industry, just taking over and taking the reins.
Russ, I'm going to throw in a question that I was going to say for later, but as long as you brought that up, so there's a woman in our lab that's a young leader. She's going to be a superstar, and I happen to be texting her about some stuff. Um, and I said, "Hey, I'm talking to Russ Weiner later today. What do you want me to ask him? Give me any question." And the question that she wanted to know is, she said, "What advice does Russ have for me to be as successful in my career as he has been in his?"
I'd say two things. One is embrace the fear, right? You will not make it in this field unless you take a chance. You'll be happy, maybe. You'll be compensated, but to get that level of happiness and to stretch. And I remember after being at Bristol Myers Squibb for 18 years, the hiring manager at Merck said, "Embrace the fear." And I'm like, all right, I'll do it. And I tell you, I've never looked back. Moving over to Gates Medical Research Institute and not knowing a thing about infectious disease, and moving to Boston after 20 some odd years in New Jersey, embrace the fear, right? Kids are in school. You have no reason not to take a more challenging job in pharma, in not for profit. And I'll tell you, so it's embrace the fear. And the other one is never pass up an opportunity. So if your boss says, "Hey, what do you think about this?" You don't go, "Ugh." You go, "Okay, embrace the fear and take that," because that's usually going to parlay into something else. And I do it every single day. If there's a meeting that's taking place, or I have to go up on the stage and read a teleprompter, which I hate, I embrace the fear, and hopefully if I mess up, it's comedic and people will enjoy it.
Do you hate the teleprompter, or do you just hate? You don't hate public speaking, right? Because
No, no,
no,
you
it's
can't
the
get me to shut
teleprompter,
up.
right? Yeah, yeah. No,
I,
I'm the same. Yeah. Okay.
Yeah, the joke I used to say is when I went to grad school, I was surprised they didn't make me take the TOEFL exam. You took the GREs, but
Right.
I always done so poorly on standardized tests that they were in their right if they wanted me to take the TOEFL exam, which is, for people whose English is not their first language.
That's awesome. I love what you said there. Embrace the fear. I think that's so key to success and breaking through barriers. It sounds like our second T-shirt, Chad, because we talked last time about science is not a conspiracy theory, and embrace the fear. That's the next one on our merch list, right? Very good.
Gonna have a whole swag library that Greg and I will be the only ones buying it, but it's all right.
Right. Famous quotes on
Yeah,
BioTalk Unzipped.
yeah, yeah. That's
Nice.
fantastic. Should we talk more about Russ's career specifically at Takeda? We alluded to it a little bit, Greg.
We did. Thank you, Chad. So tell us a little bit more about what you're doing now as head of oncology and cell therapy, clinical biomarker sciences and operations at Takeda.
So I was brought in. I was fortunate enough to be given a blank slate. How often in your career do you get to come to a company, employee number one, even though it's large pharma, and recreate a department? As Chad had mentioned earlier, building is my passion. Each of the roles that I've had have always been a major build, whether it's bioanalytical, biomarkers, or companion diagnostics. I've never had a role where I've walked in and thank you. These are the people you have. This is the structure. And so the Takeda one was really, it's almost like a startup. So when I went to Bill and Melinda Gates Medical Research Institute, that was also employee one, but on a much smaller scale. They had no drugs in the pipeline. I was employee 30 something. And so there's a vacation, a honeymoon to build that lab. At Takeda, no, the group was pretty much dissolved. We relied quite heavily on other departments to help me with logistics and operations as we brought the work in. Fast forward, there's about, I think we're at 53 people in 2 years, that
Nice.
we brought in from all over the place, relocated coast to coast to come and work for Takeda. And, the one thing I went there because, quite frankly, I knew little to nothing about cell therapy aside from what they do and how they do it, but not the bioanalytical, not the clinical trial challenges. And so that was the piece. I always make sure that I'm learning something new whenever I go to the next company. And now this group is firing on all cylinders, and it is this really close knit group of individuals. We care about each other at a really deep level. And now I get to sit back, I wish, and watch everybody, do what they need to do. And again, in order to build a group this fast, people need to be comfortable with change. They need to be comfortable with being uncomfortable. And so, the vast majority of the group are there less than a year, right? I just hit my 2-year anniversary on Thursday this week. And they are going to take a chance. Some of them are junior, because I like to have that junior, that those people that really are curious and they're sponges, the future leaders of the field, and you bring them in, and they're a little scared. New processes, new ways of doing it. Well, how'd you do this before? It doesn't matter, because we're not doing it that way, anymore. So training them, and when they make mistakes, and it does happen, have their back, right? And what I always say is, if you did something, you took a risk, and in your heart you thought it was the right thing to do, and it didn't go well, the bullet comes in my back. If, however, you did something, didn't check with anybody, is a little crazy, I'll throw you under the bus, right? So it goes both ways. You will get my incredible support. I'll take those bullets, I'm riddled. But if you do something that's a little off, I'm also pretty brutal on that side, I'll be honest.
No, very good.
That's fair. That's fair.